Chapter 12, Nervous Tissue, the last of our four main types of tissue. From the chapter, you will need to take away three broad things. Understand how the nervous system helps to keep controlled conditions within limits that maintain health and homeostasis. Learn about the different branches of the nervous system. And 3, identify and describe various types of cells that are found in nervous tissue. An overview of the nervous system. The nervous system helps to maintain homeostasis, and along with the endocrine system, it helps to keep controlled conditions within normal limits that maintain health. The nervous system is also responsible for all of our behaviors, memories, and movements. Lastly, the branch of medical science that deals with the normal functioning and disorders of the nervous system is called neurology. Next, we will discuss the layout of the nervous system. Our nervous system is divided into the central nervous system and the peripheral nervous system. The central nervous system, CNS, consists of the brain and spinal cord. The peripheral nervous system, PNS, consists of cranial and spinal nerves. These nerves have a sensory, efferent with an E, and then motor, afferent with an A components, ganglia, and sensory receptors. The sensory system consists of a variety of different receptors as well as sensory neurons, while the motor system conducts nerve impulses from the central nervous system to muscles and glands. The peripheral nervous system is also subdivided into somatic and autonomic nervous systems. The somatic nervous system is voluntary and consists of neurons that conduct impulses from cutaneous and special sense receptors to the central nervous system, and motor neurons that conduct impulses from the central nervous system to skeletal muscle tissue. The autonomic nervous system, or ANS, is involuntary and contains sensory neurons from visceral organs and motor neurons that convey impulses from the central nervous system to smooth muscle tissue, cardiac muscle tissue, and glands. The somatic is for your skeletal muscles, and the autonomic is for your smooth muscle, cardiac muscle tissue, and glands. In other words, skeletal muscle, we think about moving, and therefore it is voluntary. Smooth muscle, like our vessels, cardiac muscles, like our heart, we do not think about moving, and so it is part of the autonomic nervous system. The motor part of the autonomic nervous system consists of the sympathetic division and the parasympathetic division, which will be discussed in detail in Chapter 15. The enteric nervous system, or ENS, consists of neurons in enteric plexuses that extend the length of the gastrointestinal tract. Many neurons of the enteric plexuses function independently of the autonomic nervous system and central nervous system. Sensory neurons of the enteric nervous system monitor chemical changes within the gastrointestinal tract and stretching of its walls, whereas the enteric motor neurons govern contraction of gastrointestinal tract organs and activity of the gastrointestinal tract endocrine cells. What are the functions of the nervous system? There are 3 main functions of the nervous system: sensory, motor, and integrative. The sensory function of the nervous system is to sense changes in the internal and external environment through sensory receptors. Sensory neurons will serve this function. For example, the sensation of tickle on your skin. The motor function is to respond to stimuli by initiating an action. Motor neurons will serve this function. For example, a contraction of your muscle in response to a reflex. And the integrative function is to analyze the sensory information, store some aspects, and make decisions regarding appropriate behaviors. You will have association or interneurons to serve this function. What are the parts of our neurons and how do they function? Neurons have the property of electrical excitability similar to muscle cells. Most neurons or nerve cells have a cell body or soma, many dendrites, and usually a single axon. The cell body contains a nucleus, lysozymes, mitochondria, a Golgi complex, cytoplasmic inclusions such as lipofuscin, chromatophilic substances, and neurofibers. Many of the same things you find in a normal cell. There are chromatophilic substances known as nycil bodies that are orderly arranged as the rough endoplasmic reticulum. And there are neurofibrils that form the cytoskeleton. The dendrites are the receiving or input portions of a neuron, and the axon will conduct nerve impulses from the neuron to the dendrites or cell body of another neuron or to an effector organ of the body, such as a muscle or gland. A synapse is the site of functional contact between two neurons or between a neuron and an effector cell. Axonal transport, a natural mechanism of our intracellular transport in neurons, can be exploited by certain microorganisms to reach other parts of the nervous system. Both structural and functional features are used to classify the various neurons in the body. Structurally, neurons can be classified on the basis of the number of processes extending from the cell body. Neurons are classified as multipolar, bipolar, and unipolar. Most neurons in our body are interneurons and are often named for the histologists who first described them, such as Purkinje cells, or named for the shape or appearance, such as pyramidal cells. Functionally, neurons can be classified based on the direction of nerve impulse propagation. On the basis of function, our neurons have three classifications: sensory or afferent neurons, motor or efferent neurons, and association or interneurons. So a little mnemonic to help you here is using the word same, the S and the A are next to one another, so that is your sensory afferent, and then the M and the E are next to one another, so that is your motor efferent. And here you will see our sensory neuron will carry the impulse into the central nervous system or towards the central nervous system. So afferent carrying towards the central nervous system, and efferent with an E carrying it to the effector or exiting the central nervous system. Neuroglia or glia are not electrically excitable cells. They make up about half the volume of the nervous system, and they can multiply and divide. These are specialized tissue cells that support neurons, attach neurons to blood vessels, and produce the myelin sheath around axons, and carry out phagocytosis. The types of neuroglia include those in the central nervous system and in the peripheral nervous system. There are 6 kinds of neuroglia in total. In the central nervous system, you have astrocytes, oligodendrocytes, microglial cells, and ependymal cells. This is an image from our central nervous system. On the next slide, we'll have the peripheral nervous system, but we only have 2 types, so I'll list them here. The Schwann cells and satellite cells. Astrocyte functions include microfilaments for strength, producing the blood-brain or being a part of the blood-brain barrier, regulating growth, helping to maintain chemical environments, and learning and memory. Oligodendrocytes create the myelin sheath. Microglial cells will perform phagocytosis, and ependymal cells circulate cerebral spinal fluid. In the PNS, or the peripheral nervous system, Schwann cells create the myelin sheath, and satellite cells exchange material between neuronal cell bodies and interstitial fluid. Here are the neuroglia of the peripheral nervous system, the Schwann cell, and the myelinated axons. Myelination of neurons. Myelin sheaths are a multi-layered lipid and protein covering surrounding the axons of most neurons. This myelin sheath covering is produced by Schwann cells in the peripheral nervous system, creating the neurolemma, and oligodendrocytes in the central nervous system. This myelin sheath electrically insulates the axon and increases the speed of nerve impulse conduction, therefore allowing the nerve impulse to travel faster. The outer nucleated cytoplasmic layer of the Schwann cell, which encloses the myelin sheath, is called the neurolemma, or sheath of Schwann, and is found only around axons in the peripheral nervous system. The neurolemma aids in regeneration in injured axons by forming a regeneration tube that guides and stimulates regrowth of the axon. We will later talk more about regeneration. The myelin sheath has gaps called fibril nodes or nodes of Ranvier along the axon. Oligodendrocytes will form myelin sheath for CNS axons. No neurolema is formed in the central nervous system, and no regrowth after injury will occur. Collections of nervous tissue include several structures here. We have clusters of cell bodies that will be ganglia and nuclei. Ganglia will be located in the peripheral nervous system, while nuclei are collections or clusters of cell bodies in the central nervous system. As you will begin to notice, similar structures can have different names dependent on nervous system location. Nerves versus tracts. Nerves are groups of axons located in the peripheral nervous system, while tracts are groups of axons located in the central nervous system. Our gray and our white matter. Gray matter will contain three main things: nerve cell bodies, dendrites, and axon terminals or bundles of unmyelinated axons and neuroglia. The white matter will contain or be composed of aggregations of myelinated processes. So white matter contains myelinated processes, and that's how it gets its color, whereas gray matter is going to be unmyelinated. We show here that in our spinal cord versus our brain, the location of white matter and gray matter are different. In the spinal cord, gray matter forms an H-shaped inner core surrounded by the white matter, whereas in the brain, we have a thin outer shell of gray matter covering the cerebral hemispheres. So, in other words, the spinal cord and the brain have reverse arrangements of gray and white matter. We will pause here, and you have now been through the nervous tissue and the functions of the nervous system.